Hello, and welcome to this week's edition of EcoEvo Chronicles. This week, we're going to talk about a more recent paper that came out of the journal called New Phytologist. And this paper is called A Test of the Grant-Stebbins Pollinator-Shift Model of Floral Evolution. The paper came out of a group out of South Africa that's doing really interesting work on a group of lilies, and we'll get more into that. But first, what we should talk about is what is the Grant-Stebbins pollinator-shift model? It was first published in this chapter of this book called Ecology and Evolution of Flowers. And the chapter is called Pollinator-Driven Speciation in Plants. Basically, this person named Steven Johnson has combined two theories from really amazing evolutionary botanists named Vern Grant and also his partner named Karen Grant, as well as Stebbins. And both of these are regarding how pollinators can apply selection to plants and enforce evolution of floral form. And so, as I mentioned before, we have here Vern Grant and Karen Grant, as well as Stebbins. And so these guys are evolutionary botanists, and they focus on plants because it's really amazing how pollinators, all different types of them, can apply selection pressures to subsequently cause evolution of plant form and function. Vern Grant really focused on the fact that the quantity of pollinator visitors might determine whether a plant evolves to a different form. Let's take this example that I have here in the bottom corner, where we have this pink flower, and let's say it's visited both by bees and butterflies. Well, let's say in a subsequent generation of this flower that instead of bees being the predominant pollinator here, it turns out that butterflies actually visit this plant more frequently. In subsequent generations, we might see that this plant begins to adapt to butterfly pollination. So it might start displaying different traits, and those traits would be more suited or better suited for butterflies to visit rather than bees. To reiterate, the grants really focus on the quantity of pollinator visitors. So in this scenario, if butterflies are visiting this plant more frequently in larger numbers, in subsequent generations, you might see that this plant has evolved to butterfly pollination. Whereas Stebbins, our other character in this story, focuses on quantity. He says that's great, but we also have to focus on the quality of the pollinator visitors. So he's saying here that if we again have this scenario of the bee and the butterfly that are both visiting this pink flower here, and it might be a large population of them, we need to see that in order for this plant to subsequently evolve more butterfly preferred traits, we need to see that the butterfly is actually a better pollinator than the bees. One way that we would see this is that this plant, when it is pollinated by the butterflies, is actually producing a higher seed set when pollinated by butterflies versus the bees. So, bringing this all into the context of combining these two theories that focus on quantity and quality of pollinator visitors, Steven Johnson combined these theories and said, okay, let's say that the Grant-Stebbins pollinator-shift model is when there is variation in both quantity and quality of pollinator visitors across space. And if this happens, this will lead to the formation of plant ecotypes. There will be certain plant populations across a landscape that are more focused on getting visits from bees, while other populations are more focused on getting visits from butterflies. So that is the Grant-Stebbins pollinator-shift model. This paper that we're going to discuss today is really focused on determining whether their system of really pretty lilies is actually a good display of this model. I'm going to jump right into their first figure because it's more of a conceptual figure that again just talks about this model that they're trying to prove is or is not within this system. For their system, we're in South African plant called Brunsvigia gregaria, and it's commonly called the gregarious candelabra lily. These are two images of it. And basically, what they're trying to show us here is that there's actually some ecotype separation in this plant species. So the pink represents plants that have short styles. So a style is a female reproductive structure within flowers. Whereas this light blue notated plant here has long styles. You can see that even the flowers are larger. And so the idea with this map here, this is the whole plant range. But they say within this conceptual figure caption here, this is figure 1, that we have this plant range. These honeybees are present across the entire landscape, so both over here as well as over in this blue area. However, the butterflies are only present in the blue area. What they're trying to describe here is that there are different ecotypes of this plant based on the pollinators that are present. The pollinators over here in this pink area are mostly bees, and the plants are here within these little squares, and they're pollinated by bees. So they have pollinator and plant matching. Small bees here, like these honeybees, are able to touch the male and the female reproductive structures. Whereas over here in this blue area, the bigger butterflies are better suited for this flower type because the styles are longer. So we need a larger bodied insect in order to touch these reproductive structures back here. And one thing that they're showing us here with these squares that are shaded out is that even though honeybees are present across the entire range, they are not as efficient of pollinators when it comes to the large or long-styled plants. In the long-styled populations, the butterflies are the more efficient pollinator, therefore leading to a higher fitness. Okay, so that's figure 1 within this paper. So just to go over the aim of the paper, they're using this lily system called Brunsvigia gregaria to test the Grant-Stebbins model of floral evolution. And to complete this aim, these researchers are quantifying floral traits to determine if floral variation across the landscape exists, as well as watching pollinators to see if different floral traits are resulting in different pollinator visitors. So let's get into the figures to see what they find. Within this paper, figure 2 is showing us these wonderful plants. They're very pretty. And here, panel A is showing us a long-styled plant. It seems that this plant just grows right out of the ground and produces many of these flowers. And again, we can see right here this long style, as well as an insect that is coming to visit. Here in panel B, we have a short-styled plant with a honeybee, and we can see there's pollinator matching because as the pollinator moves backwards out of this flower, this honeybee is actually depositing pollen on the tip of the style called the stigma. In panel C, we have another long-styled plant. You can see it being visited by this fly. And in D, we have actually an intermediate length styled plant being visited by a butterfly. And then again, in E, we have a long styled plant. And we can see here that this tiny fly and this long styled plant, the tiny fly isn't matching up with the reproductive structures. So the anthers that hold pollen are way back here, as well as the style. So this is a case of pollinator mismatch. Okay, and then their figure 3, they're showing us that actually in this system, most of the time, there is pollinator matching with the plants and flowers that they are visiting. So here all the way to the left, there is a photo of the short-styled plants, and these two are the short-styled populations. We can see that amongst the ecotypes here, our green short-styled ecotype has the shortest styles of all of the populations. Then also the length of the floral visitors shown by each of these different symbols is the smallest as well. So what we can take away from this is that in each of these different style lengths, ecotypes, so short styled, intermediate style length, and long style length, there is mostly pollinator matching, where pollinator body length matches with the style length exhibited by these flowers. So next up, they looked at flower color across the different ecotypes, and they did this by using a technique called reflectance spectroscopy. And so in this technique, they are basically showing their sample. In this example, which is not from this paper that we're discussing today, it came from a separate paper. It is the strawberry, but in these authors' instance, it would be the flower tepals. And then the spectrometer takes in this information and basically outputs a wavelength curve showing the flower color in nanometer space. So what they're doing here is they're actually putting their data into some models of both honeybee in A and C panels and fly in B and D panels vision. And so the way that they're doing that is they're measuring the flower color and then they're putting this into a package in the R software, and they are trying to see in A which of the different cones of the honeybee shown here at each of the tips that are labeled are being quote unquote excited. And so they have these colored by ecotype, and you can see that there's a little bit of variation between the ecotypes. And here for B, this is the fly vision. The same is also true. Then look to see which of the flower colors is visible to bees. And in panel C, anything that is over the dotted line is visible or detectable to the honeybees. And in D, any points that are over the dotted line are detectable to the fly. So we can see that color is slightly different across the floral ecotypes. Next, we have figure 5, which is coming from their pollinator observation data. And so they're basically building these pollinator assemblages. This shows us that each of the different style length ecotypes is being visited by different pollinators. They all have different pollinator importance, which is shown in panel B. The yellow populations here are those with the long styles, and they're visited mostly by these carpenter bees and long proboscis flies, whereas each of the intermediate populations are visited by skippers and swallowtails, while the short-styled ecotypes are visited by 3 different pollinators. But we can see that there's hardly any overlap in pollinator importance, indicating that there are different pollinators that are visiting the plants based on their ecotype and location on the landscape. Next up, we have figure 6, which is showing us again some data that's been collected on the pollinators that are visiting these plants across these 3 different ecotypes. And we can see that in A and B, when comparing the weighted mean pollinator body length, as well as the body length of the most important pollinator group to the style length, both of those have strong positive correlations, which indicates that at each of these different ecotypes, there are different pollinators that are visiting each of these ecotypes. And then we also on C and D have the probability of those pollinators contacting the styles of the plants. So remember, these styles differ in length based on ecotype. This is just really good evidence that there is actually selection from the pollinators on each of these different ecotypes. Finally, we have figure 7, which is actually quantifying selection on the different ecotypes. So the colors here in this panel are determined by the number of seeds that are set by the plants in the short-styled populations. On each of the axes, we have the standardized flower number as well as the standardized style length. And we can see that there is positive directional selection on the flower number, as well as stabling selection on the style length. And then there's also correlational selection on flower number and style length. So that means that each of these traits, flower number and style length, are correlated with each other. And then in panels B and C, we're looking at the intermediate style pops, where we see positive selection on both flower number as well as on tepal length. And this is determined by, again, the number of seeds that are set. And this comes through because the pollinators are more effective at pollinating those plants and therefore producing seeds. In conclusion, within this really cool system, Brunsvigia gregaria, these researchers from South Africa found that the floral system does seem to follow the Grant-Stebbins pollinator-shift model. There are indeed multiple ecotypes across the South African landscape of this plant, the short, the intermediate, and the long styled ecotype. And each of these three different ecotypes has its own set of floral traits as well as its own set of important pollinators. And the pollinator matching between the pollinator body size as well as the style length for each of those ecotypes allows for the application of selection pressures on these plants. Thanks again for listening, and thanks to these researchers for putting out this really cool paper in New Phytologist. And if you're looking for the citation for this paper, you can find it down below in the description. Thanks, and see you again soon.